Joining us now is Dr. Anna Bento. She is an assistant professor at IU School of Public Health in Bloomington. We're always grateful to have her time and her expertise to weigh in on something that is kind of a little bit controversial, but we want to sort of untangle this concept. So, Dr. Bento, welcome. Thank you.
Thanks for having me.
So, Dr. Bento, we're talking about what's called gain of function research.
Yes.
First, would you explain what that is and what the purpose is?
Yes, of course. So gain and loss of function experiments or research is actually a host of different types of experiments virologists use to understand the genetic makeup of viruses and the specifics interactions between viruses and hosts. And hosts here can be, of course, humans, can be animals, and so on. So the idea behind the gain of function research refers to a thing we call serial passaging of microorganisms. And I'm going to tell you exactly what that means. So you kind of use several generations of these pathogens or these viruses. And then what you want to do is look to see if there's a selection for particular genes that are lost or that are gained, and how that might affect an increase in transmissibility, an increase in virulence, an increase in the ability for a virus, for instance, to be able to be better transmitted between humans or between animals and humans.
That is so interesting, Dr. Bento. So from what I read, and please clarify this, it's yes. That it's a, I was looking at the NIH website. It's been something that's been studied in the US across many institutions. Clearly, it's a safe way in which we do science, and it has a purpose, as you explained. The purpose, from what I'm hearing, is to increase the transmissibility and contagiousness of the virus, maybe for worst case scenario. I don't know. That was just from my understanding.
Exactly. So it's not always to increase virulence or increased transmissibility, is actually to try and understand the mechanisms as to how a virus can become more virulent or more transmissible. So that's why normally this type of research is not just gain of function, but also loss of function, right? So these experiments, just to put it very simply, are fundamental to understanding the biology, the ecology, of these viruses, and to actually adding basic knowledge to, for instance, what we're experiencing now with the SARS-CoV-2 pandemic, right? So the more we understand how viruses can lose function or gain function, the better we are actually at designing vaccines that will deal, for instance, with new variants that we know are emerging in many parts of the world, including the US.
That's incredible, because there has been a lot of things in the news that have been like, this is not a way, or it's very confusing why we're doing this. But you said gain of function, but loss of function, and it's how we can really attack a virus, or to be able to help us get rid of it, eradicate it. Yes. Would you expand? If that's what I'm hearing?
Yes. So I can give you an example. For instance.
Please.
So if you think about, let's step away from SARS-CoV-2, from COVID-19, and think about influenza, so the flu, right? So many years of research, and we know like with COVID-19, the flu every year has different strains, right? That's why we should get vaccinated every year, because new strains appear every year, right? So a lot of the gain or loss of function experiments that are, of course, not done in humans, but done with animal models in a very, very particular and secure setting in laboratories. For instance, for the influenza virus, there's a lot of work being done with ferrets, where it leads to the characterization of potential new variants that can pop up in the next year. So it's also a way of us to try and understand what is the probability or the likelihood of a new variant to occur in the next year or in the next season, right? So it allows us to understand exactly what can we expect. So therefore, we can design better vaccines to make sure we can control whatever new variant is popping up in the population.
science is so incredible. So are we taking the virus, or are we making a synthetic form of it, or are we taking the actual pathogen that's circulating?
We take the circulating pathogens and then we use animal models with that particular, so it's not a synthetic pathogen at all, right? We take the existing pathogen, the existing virus, and then what we do is that we infect, well, I don't, but like the virologists infect a particular animal host in a lab setting, so not humans. Let's be very, very clear here. So imagine ferrets, mice, and then they infect one ferret or one mouse, and then they look at the transmission events, right? One ferret transmits to another ferret and another ferret. So as you follow the transmission chain, which you can actually know exactly when it's happening and who is infecting whom, you are able to take out the virus, sequence the virus, and check what types of genes were lost and what types of genes were gained as each transmission event occurs, right? So it's kind of like contact tracing done in a lab setting and trying to estimate what happens every time there's a transmission event. How quickly does the virus change? And what are the types of genes that are under selection as we have transmission from one individual to another?
That, I mean, and so we're saying, I mean, from what I understand, if we infect, let's say, in animal models first, we watch in terms, I mean, are they put in sort of a close setting and then seeing the
so these are called a BSL-3, very secure type of laboratories that very few people have access to. They have to go through extensive training, and the likelihood of a leak is very close to zero. So these viruses are very unlikely to get out of the lab, or even, you know, an event of a spillover from a lab is very unlikely. So this is extremely secure and is never done on humans, is always done on animal models that have been particularly bred for these type of experiments.
Right. Dr. Bento, I know that there's been a lot of confusion in the news as whether this was something, I mean, maybe it could have helped with the coronavirus. I don't know the variants, and if we had, I don't know if we've tested this in animals and have done that type of research with the coronavirus. If we had done that, do you think that we could have, I don't know, seen these variants coming? Would it have given us a sense of variance?
So that's really hard to explain, to answer in a yes or no fashion, right? So, but what I can tell you is that there's a setting and a purpose for all of these experiments. So there will be labs doing these types of gain or loss of function for SARS-CoV-2 eventually, because these have been done for influenza, these have been done for HIV, these have been done for measles virus, and so on, right? The thing is that with the SARS-CoV-2, because we're kind of accruing information very quickly, these actual experiments take a long time to be approved. So eventually this will be done, because there is a probability that COVID is not going to go anywhere, right? It's going to probably become a seasonal problem. So there will be more and more research in terms of vaccine development done for the next years to come. And so very likely some of these experiments will eventually be approved to study precisely this idea of selection of more virulent genes, or more pathogenic genes, or more transmissibility. But it hasn't been done not because it's not safe. It hasn't been done because we just haven't had the time as a community to reach that timeline, right? There was a race to develop vaccines to control the first fire we needed to put out. We are now controlling that fire. And then we have to kind of go on to the next steps, which eventually will include some of these experiments. But again, in very few labs in the country, very few labs in the world, with animal models that never left the lab, never will leave the lab, and don't even exist in the wild.
Right, right. Because I know also there's some sort of rumors circulating or confusion that this was happening in a lab and that this COVID was released in an intentional way, which we spoke about, which you had
That's right.
traced.
This is
You
definitely not the case.
you had traced so specifically and I don't even know the word. It made so much sense what you said before,
Yeah.
and I'm going to tell I want to ask our audience, our viewers, our audience to go ahead and re-listen to the podcast
Sure.
that in terms of your explanation of that. So it just it's not here yet currently being tested on COVID, but because there's the future of COVID potential for it being a
Yes, it's
seasonal
very
thing.
likely that at some point will be something that needs to be done simply to better understand these mechanisms, right? So we had to do a lot of things at the same time to be able to control this pandemic. So this was a once in a lifetime event up to now, right? So this was the worst thing that has happened globally in centuries. So the type of research that was done up to this point was just kind of not necessarily looking to the next 10 years, but looking at the now. How can we control this now? And then as we know more, we'll have to try and understand these basic questions about the virus. What are mechanisms? How will the virus evolve with time? And these are types of experiments, not the only ones, but these are very well-defined experiments that really study these types of selection events.
Wow. Wow. And again, like you said, it's a small number of institutions.
That's
it is
correct.
so secure.
Yeah.
It is not something that is going to, escape a lab that has
Definitely
essentially
not.
it's like a bank lock. You kind of lock it in there, and
That's
8
right.
times to get into like the whatever $1 million in there.
Yeah. So if you and I were to try to enter a lab of that type of security, we would not even be able to get to the outside door because we do not have the proper clearance.
Dr. Bento, wow. I mean, that's incredible. I mean, thank you so much for your time. I always enjoy talking to you, and I really think this is going to give, a lot of clarity, a lot of, dispel some of the rumors that might be circulating, and I really enjoy it. I really enjoy talking to you, and I just want to thank you again. All right, well, you take care then, okay? Talk to you soon.
All right, bye. It was a pleasure.